Hello, welcome back. Hopefully, you're doing okay after that long video of animal virus replication. But that really is some of the funnest topics. Um, really seeing how viruses flip what we normally understand as our central dogma kind of on its head. When I first learned about retroviruses, it just kind of blew me away. So hopefully, you are also excited about that topic. Send questions if it gets a little bit confusing because I know there's lots of new terms and things. So all right, so what we're going to do next, we're going to move on into viruses' role in cancer and how we culture viruses for research and vaccines and things like that. So we're going to first start by taking a look at their role in cancer. So just a couple terms that are in the notes here. Cancer really only affects multicellular animals. It's not something bacteria have to deal with, obviously. But whenever we are a multicellular organism and our cell cycle regulation gets out of control, we produce these large masses of cells that aren't really super functional. And so this is what we call neoplasia, is uncontrolled cell division, and it results in a tumor, which is a mass of these neoplastic cells. And not all the time does the tumor metastasize, which means goes into other parts of the body. And not all the time is a tumor a cancer. Sometimes it can be benign growth. But either way, viruses can play a role in upsetting that regulation of the cell cycle. So this idea is what's called the oncogene theory. Now, what is an oncogene? So you might have heard the prefix onco in reference to like oncology, like the study of cancer, and that's where that root word comes from. So oncogenes are genes that are found in eukaryotic multicellular organisms that, when expressed, have the ability to disrupt cell cycle regulation, like mitosis and the cell cycle. So we all have these genes it's part of our human genome, but for the most part, they are kept in check. So this guy here, it's called a proto-oncogene, which means it is an oncogene, but it is repressed. It's in the repressed state. And there's a couple reasons why it's repressed. One is there's an actual gene for a repressor that blocks that. So kind of like when we were studying our operons, where we had the repressor would block the operon, and so the DNA polymerase or the RNA polymerase could not translate it or transcribe it. So proto-oncogenes usually lack promoters, and so, or sorry, they have a repressor binding, blocking the transcription. And the other reason why it's called a proto-oncogene is it lacks a promoter. So there's really no binding site for RNA polymerase. So those proto-oncogenes are, for the most part, they're turned off and they're pretty much ignored since there's nothing for the RNA polymerase to land on. So here they're saying if you have successful transcription translation of your repressor, you get a repressor, it blocks, and no cancer. That proto-oncogene stays a proto-oncogene and it's not going to be a problem. Now we have these two hits because the hits are the things that are going to mess up this regulation. So the hit might destroy the repressor or it might add a promoter. So in this first case, in this particular example, we have a promoter region that's being added by a virus. So it might be one of those retroviruses or some kind of other virus that can insert its genetic material into right in front of that oncogene, and it acts as a promoter for a place for RNA polymerase to bind. But if the repressor is still repressing, you're going to get no cancer, because even if the RNA polymerase can bind, if it's being blocked by the repressor, it's not going to function. But if you get a second hit somewhere along the line, say another virus inserts a gene or some of its information right into the middle of that repressor gene, the repressor gene is now mutated. It can no longer be transcribed and translated. Now you have a promoter and you have no repressor. So your oncogene gets expressed and it causes a protein that somehow disrupts that cell cycle regulation. You get uncontrolled cell growth, and this is what we call cancer. So there are some viruses that are really prevalent and good at their ability of doing these hits that are going to impact the proto-oncogenes kind of subdued state. Or environmental, just kind of going off a little bit, it could be a combination of an environmental exposure and a viral exposure. So there's no rule to say it has to be viral both hits to activate one of those proto-oncogenes. All right. Okay, so that's just our little conversation on viruses and cancer. Then we're going to go into viruses, culturing viruses. So if you are a virologist or somebody who's studying viruses, you need to grow viruses. But you can't just grow just a virus because they don't work that way. They have to have their host cell. So if you are studying bacteriophages, you need to grow bacteria and infect them with bacteriophages, and then you can study your bacteriophages because they're growing in your bacterial culture. So usually a lot of times you'll see them on petri dishes. This is all bacteria, but then these little circles, these are called bacterial plaques, bacteriophage plaques. That is where the bacteriophage has killed all the bacteria in that little circle. That's why there's hollow spaces. There's open little holes because the bacteriophages are there actively killing the bacteria. So this is just one way that they can say, yes, I have bacteriophages in my bacterial culture. Animal viruses clearly cannot be grown in bacteria because they won't infect bacteria. They have to be infected into an animal. So embryonated eggs are a good compromise because who's going to volunteer to be the incubator for animal viruses? Not me. But so this picture is just showing you there's various places where parts of the embryonated egg can be used to culture the virus. So it can go into the yolk sac, the actual embryo itself, the amnion, the chorioallantosis, or the chorioallantoic membrane. So sometimes you might, if you've seen the inserts in vaccines or whatever, it says if you have an allergy or sensitivity to egg protein, avoid this vaccine. It's because the viruses to make the vaccine were probably cultured in egg and then harvested, and it's almost impossible to filter out all of those egg proteins when they are manufacturing the virus. So that's one of the reasons why, if you've seen that before, that's why it's not just some weird question about if you're allergic to eggs or not, because the virus was probably grown in eggs. But again, this isn't going to work for all viruses because some human viruses will not grow in a chicken egg. Those are pretty different animals between a bird and a mammal. So the next best thing is tissue cultures. So these are cells that are susceptible to the viruses that are able to allow the researcher to infect these human cells in these cell cultures to then grow and study the viruses. Because again, who's going to want to infect a human to be a viral incubator? It just is not ethically sound choice, which is good. We don't want that to ever be a thing. So this is a way that researchers can grow human viruses in human cells in a ethical way by growing these in cell tissue cultures. All right, so this was a short video. So that's to compensate for the longer animal virus replication. So we talked a little bit about how viruses play a role in cancer by activating those proto-oncogenes, and then just the various ways that viruses can be cultured in different organisms. Okay, we'll see you next time for viroids and prions. Bye.